Welcome to Akhand Vidyashram, the first institute of incomplete wisdom. Today we are going to talk about red blood cells and 10 stages of formation. As a result, entire DIVYANK concept can be seen in red blood cells itself. It starts as a living cell and becomes a functional cell in 21 days. You are going to see many unique things which red blood cells actually show in universal consciousness and individual consciousness. Now, I said the unique red blood cell. Brain cannot function without oxygen. For that matter, every cell starts with aerobic metabolism only, and up to 61.803% it is aerobic or oxygen consent. Beyond that, it becomes anaerobic for another 38.1966% when oxygen does not play. When oxygen plays, it converts into pyruvic acid. When oxygen is not there, it is lactic acid with a conversion of the whole biometabolism. Now, for all the 100 trillion cells of the body, oxygen is absolutely essential. And that is what it is all about. Oxygen is absorbed by the haemoglobin is the second thing which I am making it very clear. Haemoglobin has the power to absorb oxygen and carbon dioxide, and exchange of gases, or rather oxygen and carbon dioxide, takes place of haemoglobin, and hemoglobin is in RBC. That's it. Second thing, a germ cell, a stem cell from which totipotent cell red blood cells are formed are 21 microns, is again interesting. 21 microns. The size increases to 21 microns in the first stage of creation, is again Brahmank. Then there are 10 stages of formation, that is Dasank, and 10 stages of development. And finally, a mature cell is 22 by 3 is Shivank into that. And 22 by 7 is pi. 22 by 3 is again little more than 7 micron is the size of diameter of a mature cell, whereas functional cell or germ cell is 3 times more. You are going to see many of these things which will reveal how beautiful is the science to be understood with spiritual science together, and I am making it very clear. Spirituality and science should go in hand in hand because they know the truth, not keep them as separate. When together only we wisdom. As on today, the modern science is deficient because there is no spirituality, and spirituality is deficient because there is no medicine, no science. Without divine mathematics, divine physics, divine chemistry, and divine medical sciences, spirituality has no relevance at all. That is my point. Now comes the question. We started with the human cells and divine design, which I started. The new series is the new things for the entire world to understand the concept incompletely. I am not going to give a sectarian thing which books talk about it. Let every child from the beginning itself understand things very clearly. For that matter, when we say arteries, all the books say arteries carry oxygenated blood, which is not true. Arteries carry blood from the heart to the periphery and veins from periphery, and oxygen or no oxygen has nothing to do with arteries and this. And this is also a correction which I would like to make as we proceed. Now this applied spiritual science is something which I have been saying very clearly. It is the DIVYANK ratio, DIVYANK constant, DIVYANK infinity, DIVYANK sequence, and all the concepts in DIVYANK only explain how the entire universe is created so perfectly from highest to the lowest. You are going to see more and more examples, including up to atom, elements, and molecules itself. Now, I have been saying very clearly, the prime objective of human life or all the life is to lead a perfectly healthy life, free from physical diseases, perfect emotional stability, material wealth and prosperity, right education and knowledge, abundance of impeccable wisdom, genuine respect for the world religions, and genuine commitment to social health, spiritual evolution, enlightenment. This is a total thing which human beings can do because we have the facilities to understand perfect anatomy, physiology, biochemistry, psychology. We can understand the entire universe because we have the great human body, great brain, great mind, great consciousness, all the divinities within us itself. No other being in the world is even half as good as a human being. The reason we fail because we do not attain, because we are ignorant, inertia, we are not working. And I am making it clear. All our faults of every human being is not because of somebody else. It is our own fault because we are not entrusting our own self. We just want name, fame, everything can buy, nothing can. Health has to be done by the person with the knowledge, wisdom. Impeccable wisdom to holds only then. Once we have the perfect knowledge of human anatomy, physiology, biochemistry, psychology, only we can be perfect. Only then we can be a walking, talking encyclopedia and live a beautiful life. And that is objective of my life as such. And I have learnt in the last 70 odd years plus how to take care of myself by myself. And again, I am not a democrat, I am not a diplomat, I am not a dictator, but I am an autocrat, self-made, self-sufficient in everything, where I have learnt how to understand self within me and learn everything how it works within me. And that is what I am trying to share with the world. If I can do it, everybody can do it. Now come to the question of, and I'm making again and again emphasis is Ganit Vidya, a divine mathematics, the only thing which brings perfection, and you can see the whole universal consciousness centers around DIVYANK alone. Now come to the cell part. Cell, if you look at it, it exactly looks like zero. Whole thing starts from zero. If the structure of cell, technically speaking, is 22 by 21 only, you can see it very clearly. And it is made of three parts. And that's what I say, Brahma, Vishnu, Mahesh, or Saraswati, Mahalakshmi, Mahakali, are everything made of three parts. And that is what I am trying to say very clearly. Design everything from 0 to 1 to 3, 5, 7, 10 and thereafter. And that is divine mathematics, that at different level how it works. So here human cell, as told you, cell membrane, cytoplasm and nucleus are three things which we see under a compound microscope. When we see under electron microscope, speed, each cell membrane is made of 7 parts, cytoplasm is made of 7 parts, nucleus is made of 7. There are only 21 parts you can see very clearly in a cell. So starting cell it is of 21 parts, and I think that is clear. Earth is made of 7 parts, Antriksh is made of 7 parts, and there are 7 heavens. Even the solar system is made of 21 parts only. Now come to the red blood cell formation. And this particular picture will say clear how from the beginning, from the time of conception to move on slowly, how red blood cells are formed. In this picture I made it very clear. Intrauterine red blood cell formation is like this. First stage, mesoblastic stage, called nucleated RBC. Here you have a nucleus and RBC both together in first 3 months of pregnancy. Then mesoderm yolk sac is an intravascular erythropoiesis. First is a mesoblastic. Second stage is hepatic. It takes place in the liver, and it is also nucleated RBC. And this is mesenchymal blood vessels and tissues are found. RBC is found in blood vessels and tissues as such. Third stage of pregnancy is a myeloid stage which means bone marrow structure. From liver and bone marrow is just non-nucleated. The nucleus is removed. First two stage nucleus is there. Third stage nucleus is removed, and from vascular stage to the liver to the bone marrow, and from complete to slowly start entering into the middle part as we grow older. Adult phase, myeloid non-existent, from peripheral to axial bone shift is seen, and that is pictured very clear. And how beautifully it goes. And if you see the graphs in this particular thing, they statistically you can see same DIVYANK there alone. How it starts from 0, goes up, comes down, goes up, comes down, goes up, comes down. That is very interesting part in DIVYANK statistics here too. Now come to the cell formation, and then I said 10 stages. The process of formation and maturation is called erythropoiesis, and then differences: proliferation, differentiation, maturation, release, functional RBC, senile RBC. Whole cells live for only 120 days. And how I am bringing this whole formation of cell takes place 21 days only, not 20 or 22. That also is very clear how divine structure. As a matter of fact, I remember as a child I saw a movie called Amar, where hen is sitting 21 days and giving an egg, and I think that was a significant point. Even different sources of information, provided we are listening carefully and looking for something unique, I think we learn quite a lot. These are 10 stages we are going to see as we move on from one to that. Now the first stage of creation of red blood cells is a pluripotent, mean totipotent, many cells can. The size is large, 21 micromillimeter. Shape is irregular, basophilic cytoplasm, large nucleus, limited cytoplasm, proliferation to reproduce extensively, can form any types of the blood cells, that means RBC, white blood cells, platelets, etc., etc. And original number remains intact. For future cell formation, things don't change. Same stem cells or pluripotent stems are there of 21 micron. That number change only multiply further and go on, and the number they produce is phenomenal. You can imagine 5 million cubic RBCs are found in 1 cubic kilometer of blood itself, undiluted. Imagine the amount of RBCs we have is a tremendous size by itself. Now the growth inducers are also involved in the process, and there are 4 growth inducers: interleukin 3, IL-3, and 2, 1, 3, 6, etc. I will not go into detail here, for a common man may not be able. You can see it leisurely as you go on. Then first stage of creation is 22 by 21 pluripotent cell, irregular, 21 micron, and volume is 900 cubic micron. That's again an interesting point. 21 into 20 into 1.7 is equivalent to 900. That is something which I am talking about average. I am not talking about differentiations. This is one thing which books talk about 900 cubic millimeter. That's it, and 21. Color is basophilic, man, is blue in color. Nucleus is large nucleus. Haemoglobin is not present. Functions is proliferation. The size increases to 2 micron from in the beginning. In this particular thing I am making it clear. Nucleus is there, and so is centrosome. So centrosome when become 1 to 2, sizes increases, and that's it. The second stage of development is committed progenitor. Size is irregular, shape is irregular, size is 22 micron, cytoplasm is again blue, nucleus is 12 millimeter in size, in micro, and hemoglobin is not present. Functions are differentiation, proliferation to differentiation, and size increases to 21 micron in this particular stage only. Third is burst forming erythrocytes. Size is irregular, 20 micron, shape is again irregular, cytoplasm is blue, nucleus is active mitosis. Now nucleus has started breaking more and more. That means DNA is going more, further division, and more. So that more development starts taking. So differentiation starts coming from regular to circular as we move. There is something new hormone is coming together, is erythropoietin, and erythropoietin is produced by the kidneys as well as the stomach, and that is absolutely essential for formation. That way, normal hydrochloric acid in the stomach and having perfect kidneys is absolutely essential for RBC, because kidneys can differentiate whether oxygen content is correct or not correct and produce erythromycin in poietin. In case RBC comes down, to produce more you need this at the third stage itself. I think that is something which I want everybody to understand. Now come to the fourth stage. Shape is irregular still. Size is 18. From 22, 21 to 20 to 18 is come down, but it's again basophilic. It's alkaline in nature. Nucleus is active again. Differentiation is going on. Proliferation to differentiation continues. Mitosis continues. That means more and more cells are being formed. And a hormone is erythropoietin, but this is a stage when two vitamins start coming into play, vitamin B12 and folic acid. These are two things that are essential along with therapy at the fourth stage to go further improvement in development itself. I think that is how it should be written and remembered. Now come to the question of fifth stage is pro-normoblast stage. Shape is again irregular. Size is 16. It's reducing now. Cytoplasm has become basophilic blue again as such, no change. Nucleus is active mitosis. Function is differentiation. Hormone is erythropoietin. B12 and folic acid continues the same thing, which in fourth and fifth is same. But in the sixth stage is a basophilic early normoblast. This is sixth stage. Shape is irregular. Size is 14. Reduce further. Cytoplasm is basophilic only. And now you can see instead of DNA, you can start RNA there. Now DNA which start cytoplasm to start centrosome, and now coming to RNA. That's it. Nucleus is active but slowly coming down. Function is differentiation. Hormone is erythropoietin, and that is ribosome synthesis starts happening in the process in which further development takes place. Now the seventh is polychromatic intermediate nucleus. Shape is irregular. Size has come down to 12. And then you start seeing multipolar color. Poly mean many colors you start seeing in RBC, which was blue to start. Now you are seeing many colors. Now this nucleus is gone. DNA is gone. Cytoplasmic division has stopped. Now comes the question of iron, copper and hemoglobin synthesis starts working from this particular stage. Hemoglobin starts functioning here. And that is why you see different colors. Once you understand that, you know this is the stage where hemoglobin is started forming. Till such time there was no hemoglobin. Nucleus has stopped. DNA is not there. RNA is very pigment. Division has started, and now with iron and copper you are going to form hemoglobin, and that's it. Now come to the question 8th. When come to 8th stage is called orthochromatic. That means it's becoming more and more of red color. Late normoblast is also the stage it is called. Shape is irregular. Size reduces from 22 to 10 micron, almost half or less than half. And now cytoplasm is becoming red in color, not a blue color. Nucleus is not present, no active mitosis and absolutely no star division. You have no further development is taking place, and now slowly you find hemoglobin increasing in size. Last it started appearing, now increasing more, and as a result it becomes red in the process. And the size and shape you can see on top as I've shown in all the 10 stages. Now comes to the question of 9th is reticulocyte stage, and shape is irregular, size is reduced to 8 micron, and cytoplasm is more red. Nucleus is no active mitosis, no nuclear cell mission, and maximum hemoglobin, and there is fine reticulum. So DNA has become RNA, has become reticulum as such. But this is also the stage, ninth stage. These red blood cells are these cells which are being formed, move from bone marrow to the blood itself. It starts appearing in blood circulation itself. So it has appeared from that ninth, goes to blood itself. It is not in bone marrow alone. I think ninth has moved out. Now come to the 10th stage. You can see the red color on top. Absolutely notice it's a mature organism. Shape, which was irregular to start with, has become biconcave, as you see in the picture. And size is almost 7, a little more than that. Cytoplasm is full red color. Nucleus is no active mitosis, and no nucleus, no cell division, maximum hemoglobin has come, full maturation taking place. Now I am bringing again a compliment, that volume of a mature red blood cell is 90, which is 1/10 to start with. Starting it was 900 cubic micron, now it is only 90, 1/10. It is 7 into 7 into 1.8. Although it was 21 into 21 into 1.7, now it is 7 into 7 into 1.8 is this. It's very, very interesting how divine has designed perfection, and I want every child, every student, every researcher, every author, every teacher, every professor of the world, every individual in the world to understand this kind of precise, concise, impeccable wisdom, not confusing thing which is given in books. All the books I have read, and I find all the books are defective, 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 and this is where I have taken a trouble in the last 30 years to experience myself and show the world where we have gone wrong with mathematics, physics, chemistry, neurosciences, etc., 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 so that there is uniform education for everyone in the world. There is no confusion, no different knowledge for poor, rich, privileged, underprivileged, rich or no difference. This is a free knowledge for everybody, and that is going to be the new impeccable wisdom for the world. This is a challenge, the whole thing. We need to move out. We used to use the artificial intelligence, all the medium of YouTube, Google and other things. This is the knowledge for everyone, and I'm making it clear. This is my life's mission, and that's it. Now I'm putting the sum up together as such. Again, summary is being presented here. 10 stages. Pluripotent 21 micron, commutative progenitor 22, burst forming 20 mol, colony forming 18, pro-normoblast 16, early normoblast 14, intermediate 12, late normoblast 10, neutrocyte 8, and erythrocyte 7. This is the complete thing you can do. From 21 to 7 is 1/3 as such. You can see those 10 plus cells in the process from totipotent to pluripotent to mature cell as such. You can see size changes from 21 to first stage, reduces from 21 to 7 micron 3 times. Volume of stem cell is 21 into 7 is 900 micron. Volume of mature cell is 7 into 90 micron. Reduction in time times volume. Shape changes from irregular to biconcave. Blue stem cells turn into red cells, and each cell gives rise to 10,000 blood cells. Duration red blood cell is 21 days. All these are not coincident but divine design. That's why the entire DIVYANK can be seen in red blood cell, and why red blood cell is especially designed an active living cell becoming a functional cell and living for 120 days, formed in 21 days. It's a beautiful work for the entire universe to see, and this is something which everybody can see because they are produced in abundance, can be studied by anybody, and I am focusing on the perfect erythropoiesis, not on defect. Let's not talk of diseases. Let's talk of perfection. Once we understand perfection, only then it is possible for everyone to be perfectly healthy, wealthy, wise and happy. And having done this, I think we are very clear to move on to the next stage of neuron cells and show neuron cells like RBCs are designed, they remain, they do not die. These cells die in 120 days. Neuron cells never die. Why they don't die? But they cannot multiply. Why they cannot multiply? Because there is no centrosome. But there is a nucleus. Here there is no nucleus, no mitochondria, nothing at all. All is disposed because it cannot form anything for further. But that has to function effectively for the entire life, which could be 100 years plus and maybe many, many mean years. But memory has to remain because every cell has, there are 100 billion neuron cells and each one can produce 20 through memory cells, and that is phenomenal. Here there is no memory, there is memory. You're going to see that as we move on. And this is the, I'm not going to talk about it. I think that's enough for the time being. This is precise, concise, but everyone should know, and let there be no deficiency in knowledge of this thing. And everyone should have this perfectly. Making it too short to 2 minutes, 5 minutes, 10 minutes does not matter. This is a perfect, at least what is required. But there is much more we can develop as we can bring everything together as we move on. I think enough for the day. Thank you very much.